let's bring on Alan Horsager. I hope I got your last name right,
It's
Alan.
Horsager, but I get Horsager, Horsager. I get
man.
many
Okay.
permutations of that name, so it's pretty good.
You are the CEO of a couple companies, actually, and you're here to speak to the launch of one of them, right? Duet Therapeutics. And so if we can, Alan, let's get your slides up. Just go ahead and share your, you can click share screen on the bottom, bring up your slides, put them in slideshow mode, and there we go.
Do you see them, Spencer?
Yes, we do.
Terrific.
All right, the floor is yours, sir.
All right, appreciate it. So again, my name is Alan Horsager. I'm the president and CEO of Duet Therapeutics, and then also the president of immuno-oncology for our parent company, which is Scopus Biopharma. I'm going to talk a little bit about Scopus in the first few minutes, but then the focus of the talk will be around the launch of Duet Therapeutics, which is a wholly owned subsidiary of Scopus. So a little bit on our disclaimers. I'll skip over this relatively quickly, just in the interest of time. So a little bit of data around Scopus. It's a clinical stage biopharmaceutical company. It is traded on the NASDAQ Global Market, and there's some numbers here around it. So the meat of the talk again will be around the acquisition and launch of Duet Therapeutics. So on September 2nd, Scopus announced the launch of Duet, which integrates technologies, management, and clinical development around their immuno-oncology efforts that were in Scopus, as well as the company that they acquired, Olimmune. And I come from Olimmune, and that was acquired in June of 2021. And so that together birthed the company Duet. So Duet is creating a set of targeted oligonucleotide therapies for treatment-resistant cancers, which include blood cancers such as B-cell lymphoma and solid tumor cancers like prostate cancer or head and neck cancers. So oligonucleotides are small strands of RNA and DNA, or. So I'll tell you a little bit about our oligo, which combines the two. So some high-level highlights of the company. One important factor is this is a pretty unique technology with a bifunctional mode of action. So we have a single oligonucleotide, which does two things. One, in one part of it, it stimulates the immune system so that the immune system is aware that there's cancer and it comes and attacks it, but it also releases this immunosuppression that occurs within the tumor microenvironment. So inside the tumor, and I'll talk about this a little bit, the tumor uses immune cells and other factors to create this immunosuppressive environment, which allows the cancer to flourish and metastasize. So we break down, we trigger an immune response to the cancer, but we also break down this barrier, this immunosuppressive barrier. And I think this is important that as I mentioned, we can target both hematological malignancies and solid tumors, and specifically what are called immunologically cold solid tumors, which are cancers that are very difficult to treat, like metastatic prostate cancer. The product that we're using is a targeted approach. So we're not delivering this, we're not bathing the entire body in this drug and hitting every cell. It's taken up by a very specific set of cells, immune cells that are in this tumor microenvironment. So we're a clinical stage company. We have a phase 1 clinical trial running at City of Hope for B-cell non-Hodgkin lymphoma, and I'll talk about that a little bit. And we also have two other programs that we're developing for an additional product that will go into phase 1 trials starting in Q1 2023. We're building we're a fairly new company, in the grand scheme of things, but we're starting to build out a really talented team, and I'll talk about a few of those members and where we're focusing energy on bringing other people in. And finally, our IP position is quite strong. We have 4 issued patents that cover the technology that have been granted in multiple countries across the world, and we also have 4 PCTs that cover the technology as well. So a pretty strong portfolio of IP. So there are 3 different technologies that we have in our portfolio. They're called Duet 01, 02, and Duet 03. And they all have a similar structure. They're all oligonucleotides, and they contain two components. And the first component is what's called a CpG oligo, and that CpG is it creates immunostimulation. And I'll talk about how it does that. So that's what triggers the T cells to come to the tumor and start attacking the tumor itself. And then the second part is this what we call a STAT3 inhibitor, and that takes different forms. In one form, it's an RNA silencing mechanism. In another, it's an antisense, which is also a silencing mechanism. And then lastly, what's called a decoy molecule, which inhibits STAT3 at the level of the protein. These inhibitors of STAT3 are what releases this immunosuppression. So in each case, we're stimulating the immune system and then releasing the immunosuppression through this secondary component. So the Duet 01, we're focusing on B-cell non-Hodgkin lymphoma, which is we've received IND clearance, and we're enrolling patients in this trial, and we're hopeful that we'll be dosing our first patient this next quarter in Q4 2021. And then for Duet 02 with this antisense molecule, that can be delivered systemically. So we can do an IV injection, as it's pretty stable in human serum, and we're going after genitourinary and head and neck cancers. So we'll be working on the preclinical development, the tox work over the next year, and then submitting our INDs to the FDA at the end of 22, and then beginning the trials in Q1 of 2023. In the decoy molecule, we have some early data. I'd say this is earlier in our development, but we have some early intriguing data around acute myeloid leukemia. So we'll be looking to potentially move it forward in that indication. So this is sort of a different slice of what I just said, but this is our overall pipeline, which we think is quite robust. First indication is in B-cell lymphoma. That's the one that's in the clinical study that I mentioned. We also are interested in how our product, our oligonucleotide, might work in combination with checkpoint blockade. So immune checkpoints like PD-1, PD-L1, CTLA-4, checkpoint inhibitors. I assume those have been talked about over the course of this session today. We're interested in how there might be synergies. So we may be looking at a secondary trial or a different arm of this first trial to look at that combination. And then there's the 2 trials that I mentioned for Duet 02 for genitourinary and head and neck cancer. And then we have a couple of earlier stage programs, one of which is that one with the decoy molecule for AML, but we're also interested in using that Duet 01 in combination with immune checkpoint inhibitors for cutaneous T-cell lymphoma. So I think it's a pretty good, robust pipeline. So I want to spend a little bit of time talking over the science, because I think if we can get you to feel comfortable with how this technology works, I think you'll really understand where we're differentiated and how we're quite valuable, I think, for the treatment and in immuno-oncology. So the area that we're targeting in oncology is what's called the tumor microenvironment. So imagine a group of tumor cells that form this ball, and then they recruit different immature myeloid cells, which are dendritic cells, if you've heard of those, macrophages, MDSCs, to this tumor microenvironment, as well as Tregs, to create this kind of shell or protective layer that protects the tumor and allows it to grow and flourish. So it sort of fools the immune system and says, hey, don't worry about it. We're all okay here. You don't need to attack us. And a secondary mechanism they use is checkpoints, right? So we know of checkpoint inhibitors. So they use checkpoints to try to evade immune attack by cytotoxic T cells. So they use these 2 mechanisms to create this TME, or tumor microenvironment, to allow the tumor to grow and then metastasize as well. And that's what we're trying to break through. We're trying to break through this protective layer and allow the immune system to ultimately attack the tumor. So this is very much an immune-driven approach. It's not targeting the cancer cells, which I'll talk about. So one of the main targets that we're going after is what's called STAT3. And so STAT3 is essentially a molecule that is involved in maintaining immunosuppression in the tumor microenvironment. It was in the normal body, normal healthy body, it's involved in wound healing and creating an environment to allow the wound to heal properly, and then it goes down again. But in this case, the tumor uses STAT3 to create this immunosuppressive environment. And it does this through various cells, like I mentioned, the dendritic cells, the myeloid-derived suppressor cells, the macrophages, which aren't listed here. It does a little bit in tumor cells as well, but these are the cells that we want to target and inhibit that STAT3 activation. So we want to knock down or inhibit STAT3. But STAT3, on the other hand, in other cells can be good. So in cytotoxic T cells, which are the cells that kill the cancer, it leads to T cell expansion and the formation of memory cells so that it can recognize that disease later on if those cells begin to proliferate through different metastases. So you want to shut down STAT3 in certain cells, but you want to avoid them in others. So this means you want to make sure that if you're delivering a STAT3 inhibitor, it needs to work in certain cells and avoid other cells. So it needs to be cell specific. And so the way we do that is through this bifunctional molecule that I've talked about, right? So we have two parts. One, which is that CpG. It's also a TLR9 agonist. Think of this just as an immune stimulator, and that's linked to this STAT3 inhibitor. And that immune stimulator, that CpG, actually serves as our delivery tool. So on the specific cells that we're targeting, the dendritic cells, the macrophages, the MDSCs, they express what are called scavenger receptors. And the scavenger receptors have a high affinity for this CpG and allows this whole molecule to get taken up into the cell. And then that CpG will then bind to what's called a TLR9 receptor in an endosome. And it binds this TLR9, which triggers this immune response. So it starts to say, hey, T cells, come to the tumor and start killing the T cells. And then what happens too, after the binding to a TLR9, that whole molecule gets released into the cytosol. And then that STAT3 inhibitor can then bind to a STAT3 mRNA, or in the case of the decoy, the STAT3 protein, and knock it down and shut it off. Right? And that releases the immunosuppression within the tumor microenvironment, allowing those T cells to come in and recognize the tumor and ultimately kill the tumor cells. So it's this bifunctionality, this triggering of an immune response and knocking down of the immunosuppression which are required. There are drugs out there that just target the TLR9 receptor. They create an immune response, but the problem is that immunosuppression is still there, so they don't work quite as well, and vice versa. There are STAT3 inhibitors which don't ultimately trigger this immune activation. So you need both, and they need to be happening in concert. And that's what we can do with our bifunctional molecule. The other part that's important that I mentioned in the beginning of this slide is that this CpG, or this component, allows for the uptake of this molecule into the cell. And STAT3 is localized on the interior of the cell, which makes it very difficult to get to. So other molecules that are targeting surface receptors, it wouldn't work for knocking down STAT3. So hopefully that makes sense. But it's if you take anything away, it's this bifunctionality that triggers immune activation as well as the release of immunosuppression that's key to creating durable anti-tumor responses. So I'm not going to go into too much detail. I just, suffice it to say, and if you want to reach out to me, please do if you want to dig into the data. But a couple things to point out. This is, a small slice of the data that was collected around using this first molecule, Duet-01, for B-cell lymphoma. But this first set of graphs, what it's showing you is that it's taken up pretty readily over time, and as a function of dose, into the dendritic cells and the B cells, which are the cells that we're targeting. And it's not being taken up in natural killer cells or cytotoxic T cells. So we are targeting exactly the cells that we want to target for the uptake and avoiding the cells that we don't want to have take up the molecule. And over here on the right, what you're looking at is tumor volume in a model of lymphoma. It's an A20 mouse model of lymphoma. And it's showing the Duet-01 molecule therapy in red compared to just that CpG molecule. That's just the immunostimulating molecule by itself, or just saline solution in the black. So you can see that over a regimen of about 5 dosings with the Duet 01, it's in combination with radiotherapy in this experiment, you see a complete ablation of the tumor. It's a complete reduction of volume of the tumor, which is sustained over roughly 90 days or 3 months. You can see that with just the CPG, it helps slow down tumor progression for a period, but then it comes right back up and starts to grow again. And of course it does with just saline solution. So the fact that we see this complete ablation of the tumor was very encouraging. And then of course, as I mentioned, there's a lot of other data that supports this, but that's what encouraged us to move into the clinic. So as I mentioned, we've communicated with the FDA and received IND clearance. And we're in the middle of a early stage of the beginning of a phase 1 clinical trial that's happening at City of Hope, that's being led by Dr. Elizabeth Buddy. And the 2 objectives that we have, of course, are determining the right dose that we'd use for phase 2 trial, as well as understanding the safety profile of our molecule, particularly at different doses. But we are looking at efficacy. How is our molecule working, both from an imaging standpoint as well as an overall survival. We're also doing blood samples periodically to measure biologic activity. So we'll look at STAT3 silencing, as well as how we're stimulating the immune system, both locally at the point of injection at the primary tumor, as well as systemically. So I'm going to turn attention to our second molecule that I talked about. This is Duet 02. This is essentially a very similar molecule, but is now using the STAT3. Instead, the STAT3 inhibitor is using an antisense molecule. And I won't go into any detail, but it's a slightly different mechanism of action. But it's highly stable in human serum, which means that you can inject it in the blood, right? You can do an IV injection. It can last in blood for upwards, I think the half-life is like 48 hours or something like that. However, antisense molecules typically can be cleared through the liver very rapidly. So you do want to see rapid uptake of the molecule. And that's exactly what we see. We did an experiment where we looked at the uptake in dendritic cells and macrophages, and using fluorescence, we see that the molecule is taken up rapidly in 30 minutes or less after injection. So that's good. So we see good quick uptake. And then further, we see that STAT3 is inhibited over the course of over days. You can see that the protein is knocked down almost to zero over the course of a couple of days. And very importantly, when we look at T cell proliferation and killing activity, you can see that with our molecule, which is in red, this is Duet 02 in red, we see a massive increase in T cell proliferation into the tumor, as well as its killing activity. So it's clearly killing cancer cells. And you can see that compared to PBS, which is the gray bars, or CPG alone, which is just the immunostimulatory component, or just the STAT3, which is the STAT3 inhibitor or release of immunosuppression component, they don't do nearly as well proliferating the cells or in their T cell killing activity. You really need the combination. And then this is survival data. So these are mice that are models of prostate cancer. So these are models of metastatic prostate cancer. It's bone localized prostate cancer, which is very difficult. They're both very difficult to treat models. We look at 2 models. One is an RM9 model, and the other is a PTEN deficient prostate cancer model. And again, both are very difficult to treat. They're hormone resistant. They're also immune checkpoint blockade resistant. So anyway, but probably as difficult a model as you can find to treat. So with our molecule, the Duet 02, it's in red. We see that it's a very rapid progression of the tumor, and these mice die very quickly untreated. But we find that over 50% of the animals stay alive as long as we measured with treatment from our molecule. And again, the CPG or STAT3 by itself didn't do much to improve above what you see with just the control of PBS. In the P10, you see even a more dramatic effect where you see upwards of 80-90% of our animals surviving over greater of 210 days. And again, most of those animals die off in the control or just the CpG or STAT3 alone. And this is data that we presented today at the Oligonucleotide Therapeutics Society, comparing our molecule relative to checkpoint inhibitors. So this is looking at PD-1 and CTLA-4 in the blue and red curves. And you can see with the CTLA-4 blockade, there's some survival, but it's not nearly the survival levels that we see with Duet-02. So we see pretty good benefit of our molecule over that of just a checkpoint molecule. And I think this is primarily due to the fact that these tumors are immunologically cold, so they don't have a lot of T cells in them to begin with. So it makes it hard for just the release of this immunosuppression to have a dramatic effect. And this last curve, I'll just touch on briefly. This is showing our molecule again in red in this RM9 model versus the two components that are separated. They're separate molecules co-injected at the same time, and that doesn't work. Right? It's not any better than what you see with the PBS. And the reason that is that CpG, that first component, isn't chaperoning the STAT3 inhibitor into the cell. So that STAT3 inhibition can't occur because that STAT3, as I mentioned earlier, right, that STAT3 target is intracellular. So it's difficult to get to. You've got to get that molecule into the cell so it can do its job. And we can do that with our molecule, but if you separate those molecules and try to co-inject them, you're not going to be able to do it. So last piece of data, we looked at this also in head and neck cancer. These are mice that have human head and neck tumors transplanted into them. So it's an SCC1 tumor. And we saw in these animals that we see with our treatment, we see infiltration of T cells. So we activate the tumor, the T cells. They go into the tumor. We do a cross-section of that tumor. We see cytotoxic T cells. Exactly what you want to see, right? You want to see those cytotoxic T cells in the tumor. That's a clear indication that the tumor is responding to your therapy. The other thing that's quite interesting here, right, is we used what are called immunodeficient mice. So these mice don't have an immune system. And then we took some of these mice and introduced a human immune system to them. So they have a humanized immune system. And so what we see are two things. One is that the tumor volume is decreased and at least stabilized, and in the humanized mice, but we don't see that happening at least to the same degree in these immunodeficient mice. So this suggests that our mode of activation or mode of action of the molecule is through activation of the immune system. We're not targeting cancer cells. We're targeting the immune system to teach it to attack the tumor. Secondly, this gives us great confidence that when we get into the clinic with this molecule, this should work. This is working in concert with a human immune system. We're working with human immune cells in this model. So it gives us a lot of confidence that this is going to be, it's pretty compelling that this is going to work well in the clinic. So we've started to think about these trials. So the genitourinary cancers, we're going to be looking at metastatic prostate cancer. As you've seen through the data, that's encouraging for us. We're also going to look at kidney cancer as well as bladder cancers. This is going to be done as a monotherapy. And we're starting to work with investigators at City of Hope, but we're going to be doing a multi-center, company-sponsored trial for these. Same thing with head and neck, but in this case, we're going to be combining Duet-02 with radiotherapy. We have some thesis for that. We think it works really well synergistically with radiotherapy. Can talk about that offline if people are interested, but we'll be targeting squamous cell carcinomas of the head and neck for that. A little bit about our overall timeline for the next bit of time, as well as our milestones. So I'll focus on Duet-01. So we're looking to ideally dose our first patient. We have to identify them. They're a tricky set of patients to find, but we're hopeful we can find one, and we'll dose our first patient in the fourth quarter of this year. And then recruit enough that we can begin to talk about initial clinical results in maybe Q3, maybe Q4 of next year for Duet-01. And for Duet-02, we're going to be completing our manufacturing for the molecule that we'll use for our IND enabling study. So this is all our tox work, which we'll do in the first half of 2022. And then at the end of 2022, we'll complete our manufacturing for the clinical study of the GMP product as well as submit our INDs for both the head and neck indications as well as the GU indications. And then in Q1, we'll begin our recruitment of patients for those 2 indications. A little bit about the team. I'm going to only be able to talk about a few people here. So I've mentioned who I am by background. I'm a neuroscientist by training, but I've spent the last 15 years as an entrepreneur and company builder, working for 2 startups, starting 2 of my own, and then working with Allimmune, with Marcin, and then obviously here with Duet and Scopus. Marcin Korluski is a professor at the City of Hope and the inventor of these technologies. He's a co-founder of Allimmune and continues to be a senior scientific advisor to the company. He worked with John Rossi and was a co-founder of Allimmune. And John is an amazing faculty member, but also an entrepreneur who's founded multiple companies, Allimmune, Minna, and Dicerna, who maybe some of you have heard of. He founded Minna with Professor Nagi Habib, who's a professor as well. And so these are kind of the early, these 2 folks here, John and Nagi, are part of our scientific advisory board. And over the coming weeks, we'll mention a number of other people that we've already brought on. But what I'd say is we're really, as a clinical stage company, being going to be focused on bringing on expertise in that capacity.
All right, Alan, we are up against time. So
perfect timing there.
Perfect. Look at that.
Yeah, look at that. Okay, so you want to break out of screen share?
That's fine. Yeah. All right. Duet Therapeutics. The URL is up on the screen for Scopus Biopharma. Alan, thank you so much for coming on and taking us home today at the Benzinga Small Cap Healthcare Conference.
Excellent. Appreciate it. Thanks for having me.
Thanks a lot, Alan.